Good day! Again, I'm Maylati Sarmiento. In today's video, we will discussing about the process of mitosis and meiosis. So let's start with cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. Cell division usually occurs as a part of a larger cell cycle. In eukaryotics, there are two distinct types of cell division. A vegetative division whereby each daughter cell is genetically identical to parent cell, or called the mitosis. And reproductive cell division, whereby the number of chromosomes in daughter cell is reduced by half to produce haploid gametes, or called the meiosis. Cell division, also known as mitosis, that take place in the regular body cells and keep cells living and growing. The mitosis is a process where a single cell divides into two identical daughter cells or cell division. There are basic phases of cell life: the interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. The interphase is the period of the cell cycle during which nucleus is not undergoing division, typically occurring between mitotic or meiotic division. The interphase is the longest stage of cell life. The time spent between division, the processes of materials produced for growth, or preparation for division. The first part in interphase is the G1 phase of the cell cycle or growth phase. Another part is known as S phase of a cell cycle. Synthesis phase when DNA duplicates. Next is the cycle is G2. G2 is just a checkpoint to make sure the DNA is correct. And the last part of cell cycle is called mitosis and has four phases during which the cell divides into two cells. Interphase is the cell spend the majority of its life here, growing and functioning during the S phase of a cell cycle of DNA replicates and anticipation of mitosis. Prophase, the first stage of mitosis, the process that separates the duplicated genetic materials carried in the nucleus of parent cell into identical daughter cell. During the prophase, the complex of DNA and proteins contained in the nucleus known as chromatin condenses. In this sample, this is the cell begins the division process. In this picture, this is the nucleus disappear and the nucleus membrane breaks apart. In the other picture, the chromosome become visible, this one. And the spindle apparatus form and attach to the centromeres of the chromosomes. Here, this one. Next, in early prophase of mitosis, the chromosome gets small. Centralize move the poles of the nucleus and spindle fiber develop. In the late prophase happen when the nuclear envelope disintegrates and spindle fiber begins to move chromosome toward the center of the cell. Metaphase is the second process that separates duplicated genetic materials carried in the nucleus of a parent cell into two identical daughter cells. Metaphase is the second phase in mitosis. In this picture, this is the metaphase. The nuclear membrane is completely gone. The duplicated chromosome line up along the cell equator. And this is the image of metaphase, and this is the spindle apparatus. During metaphase, the chromosomes line up across center of the cell, also called the equator or metaphase plate. Anaphase is the third stage in mitosis, each other the opposite ends of the cell. Before anaphase begin, the replicated chromosome called sister chromatids are aligned at the long equator of the cell, on the equatorial plane. The sister chromatids are paired of identical couples of DNA joined at a point called the centromere. And in this picture, the diploids of set of daughter chromosomes separated. In this one, they are pushed and pulled toward opposite poles of the cell by the spindle fibers. And this is the image of total spindle fiber in early anaphase. In anaphase, the chromatids that make up each chromosome move apart and travel to opposite ends of cellular spindle. And the final stage of mitosis is telophase. Telophase begin when the replicated pair chromosome had been separated and pulled to opposite sides or poles of the cell. This is the nuclear membrane and the nuclei or nucleus reform. This one. Cytokinesis is nearly completed. And this is the final look. The cell plate in early telophase. In telophase, an envelope surrounds its set of chromatids to form new nucleus and the cytoplasm starts to divide. Cytokinesis is the final stage of mitosis. The cytoplasm, organelles, and nuclear materials are evenly split and new cells are formed. And this sample of image of cell plate. The two new cells exactly like the other are called daughter cells. Cytokinesis takes place when the cytoplasm divides and two cells with identical genetic materials are formed. Going on the meiosis. Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs, and females. Meiosis 1. In meiosis 1, chromosome in a duplicate cell resegregated, producing four haploid daughter cells. It is step and meiosis that generate genetic diversity. Meiosis 1 is the first of two consecutive divisions at the nucleus of eukaryotic cell during meiosis, and composed of the following stages: the prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. The first stage of meiosis 1 is the prophase 1. The starting cell is diploid 2n equals 4, and homologous chromosomes pair up and exchange fragments in the process of crossing over. During prophase 1, homologous chromosomes pair and form synapse, a step unique to meiosis. The paired chromosomes are called bivalent, and the formation of chestomata caused by genetic recombination becomes apparent. The chromosal condition allows this to be viewed in the microscope. Metaphase 2. Metaphase 1. Homologous pair line up at the metaphase plate. In metaphase 1, bivalent each composed of two chromosomes or four chromatids aligned at the metaphase plate. The orientation is random with either parental homolog on a side. This means that there is the 50-50 chance for the daughter cell to get either the mother or father homolog for each chromosome. Anaphase 1. The homologs separate to opposite ends of the cell, and sister chromatids stay together. During the anaphase 1, the chestomata separate. Chromosomes, each with two chromatids, move to separate poles. Each of the daughter cells is now haploid or 23 chromosomes, but each chromosome has two chromatids. Telophase 1. Telophase 1 is the newly forming cell are haploid n equals 2. Each chromosome still has two sister chromatids of each chromosome are no longer identical to each other. In telophase 1, nuclear envelopes be reformed or the cell may quickly start meiosis 2. Now let's proceed with the meiosis 2. the second of the two consecutive divisions of the nucleus of nuclear cell during meiosis and composed of the following stages: the prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 2 is similar to meiosis, where, however, there is no S phase. The chromatids of each chromosome are no longer identical because of recombination. Meiosis 2 separates the chromatids production two daughter cells, each with 23 chromosome haploid, and each chromosome has only one chromatid. Prophase 2 starting cell are the haploid cell made in meiosis 1. Chromosome condense and the nuclear envelope breakdown. The centrosome moves apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. Prophase 2, these are the condensing of the chromatin into chromosomes, distension of the nuclear envelope, migration of centrosome to either pole, and the reconstruction of the spindle apparatus. However, centrosomes are not present in a cell. Metaphase 2. Metaphase 2 is the chromosome line up at the metaphase plate. This is the phase where the two daughter cells produced during the first mitotic division have their mitotic spindles start to draw the chromosome to the metaphase plate again. Anaphase 2. Their chromatids separate to opposite ends of the cell. The stage when sister chromatids of every chromosome separate and begin to move toward the opposite ends of the cell. The separation in the movement is due to the shortening of the chenophore microtubules. Telophase 2. The newly forming gametes are haploid and each chromosome now has just one chromatid. A nuclear envelope forms around each set of chromosomes and cytokinesis occurs producing 4 daughter cells, each with a haploid set of chromosomes.